Once we can make these rare sugars, we can now make what's called activated sugars, and these sugars have are basically the starting materials to be able to stick sugars onto other molecules, either making oligosaccharides, which is a string of sugars, or putting them onto small molecules like antiviral and anti-cancer drugs. To do that, you have two steps. You've got to first stick a phosphate on the sugar, and we've got a proprietary enzyme we call the zukinase. And again, here's one that we've discovered from, we've discovered a variety of these. Here's one from a thermophilic organism that works at high temperature, and this is a simple screen. The red is the presence of the sugar, and over time you can see the yellow company represents that we've made the sugar 1-phosphate, and these, depending on, we have different sugars down here, and so this enzyme works on a whole variety of sugars, and we can very rapidly make sugar 1-phosphates from a number of different rare and non-rare sugars. And again, these are not available, but they're an essential building block for making these activated sugars. And we also improved and changed the specificity again through making mutants through directed evolution applications. Here the wild type enzyme was only, of this particular enzyme only worked on one sugar. We were able to expand the substrate specificity profile of it by creating proprietary mutants of the enzyme. Okay, so we can now make a whole series of sugars, and we sell these as small pharmaceutical quantity products to the pharmaceutical industry. Okay, so you need these activated sugars to, actually I'm going to go on to the next slide. You need these activated sugars to attach sugars to other molecules. And why is that important? This is vancomycin. It's known as the antibiotic of last resort. It's a very important drug, but if you develop if bacteria develop drug resistance to it over time now, if you catch a vancomycin-resistant strain of bacteria, you're going to have a hard time getting cured. You can see this molecule has two parts. It's got what's called the aglycone, which means without sugar, and then the sugar attachment to it. And these sugars, depending on the molecule that they're attached to, and they're attached to a lot of natural products, can do everything from improve the general activity, change the host range, improve solubility of the molecule. They might be involved in tissue targeting, and of course sweetness as well too. It was found by one of our advisors who worked on this when he was at Eli Lilly that if you took the vancomycin aglycone and you were able to attach a sugar onto another position over here, you could significantly impact the host range of the drug, and you can now kill. You converted vancomycin's activity from being bacteriostatic, which means that it just prevented the bacteria from growing, to bactericidal, which meant it actually went out and killed the bacteria, and in addition could potentially change the host range, so that you could actually now kill some of these resistant bacteria.